Thank you for joining the Spotlight Theater sponsored by GenScript. I'm Claire Zhou, Global Product Manager for CRISPR Services at GenScript. In this session, we'll have two speakers, Dr. Ye and Dr. Burgess, sharing with us their research. Our first speaker, Dr. Lumeng Ye, is Senior Scientist from our R&D team. She received her Ph.D. from Free University Brussels and postdoc training at Technical University of Denmark. Her expertise is in synthetic biology, and she has been leading a team on developing CRISPR-based non-viral DNA template at GenScript for more than 3 years. With that, let's welcome Lumeng and get started on our first talk. Lumeng, please take over the stage.
Thank you so much, Claire. And I would like to thank everyone for joining our talk on discussing how CRISPR, especially Cas9 nuclease, chemically synthesized single-guide RNA, and a single-strand DNA HDR templates can be used together to generate cell therapeutics with a non-viral protocol, which can be further developed into an alternative solution to produce allogenic cell therapeutics in a more secure and economic way. Chimeric antigen receptor T cells are a promising treatment for certain types of cancers. And since 2017, there have been 4 CAR-T therapeutics approved by FDA. Meanwhile, cell therapeutics are still constantly evolving and improving and providing new options for cancer patients. Cell therapies are currently being evaluated in a variety of cancer types in clinical trials, all thanks to the continuous development of gene editing tools. There are several types of tools available for gene editing, and their invention all started in the realization that the introduction of double-strand DNA break at the target site may result in a several magnitude increase of frequency of target gene integration. However, the meganuclease approach was restricted by the lack of flexibility and the recognition site. Later on, there are also zinc finger nuclease and the transcription activator like effector nuclease TALEN successively increased the genome editing efficiency. However, targeting different sites in the genome require redesign and the re-engineering of a new set of proteins. The difficulty in cloning and protein engineering of zinc finger nuclease and TALEN partially prevent these tools from being broadly adopted by scientific communities. In this respect, CRISPR has indeed accelerated the field because it's very robust and much simpler and more flexible to use. Because the Cas9 protein is stable, and whenever we need to change a new target, we just need to redesign a 20 base pair guide RNA sequence and send it to chemical synthesis. It's very easy and simple to use. Therefore, CRISPR tools have been widely used in different research fields, from basic genetic research to gene therapy, cell therapy, which we'll talk a little bit more later in the talk, and also to cell line engineering and animal model generation, agriculture, target screening and epigenetic research, and also which were also very important in 2020 with the outbreak of COVID-19, fast and portable detect tools based on CRISPR also showed a great power to prevent the outbreak of COVID-19 virus. Also, because the wide use of this CRISPR tool, the inventor Emmanuel Charpentier and Jennifer Doudna received a Nobel Prize in Chemistry last year. Also, CRISPR has been used to generate cell therapeutics for clinical use nowadays. This table from the 2020 article entitled Gene Editing Pipeline Takes Off in Nature Review listed 12 allogenic therapeutics that are in or approaching the clinical trial. There's 1 engineered by meganuclease, which we mentioned before, 5 by TALEN, and 6 already by CRISPR-Cas system. Notably, the number 4 candidates, its performance in phase 1 clinical trial was already published in last February and proved the safety of CRISPR-engineered T-cell therapies. However, when we look at the process of this NY-ESO T-cell engineering, For generation of this cell therapy, CRISPR was only applied to knock out the endogenous TCR gene and the PD-1 component. The delivery of the payload NY-ESO was still done by lentivirus. Conventionally, we can also generate allogenic CAR-T or TCR-T cell by deliver an expression cassette of the payload first by viral methods, and then use the CRISPR to knock out certain endogenous targets which may cause life-threatening graft versus host disease, like the gene encoding TCR and other targets like CD52 or B2M to enhance the persistence. But actually, by CRISPR-Cas system, the functional knockout of endogenous TCR and the site-specific knockout of the site-specific knock-in of payload can be achieved by one step and in a non-viral methods. When designing the cell therapy production process, one important factor that I need to consider is the availability of critical starting materials. Lentiviral gene transfer is the current golden standard for clinical manufacture of CAR-T cells. High expression levels after random transgene integration are achieved by use of strong viral promoters. However, the excessive CAR expression may also lead to the activation-induced cell death or exhaustion. On the other hand, for non-viral methods, it has also its advantages on precise gene editing, and the entire production process is animal-free, and the much lower cost and the faster turnaround time because the entire process is simple and there's no involvement of bacterial cell or mammalian cells. During the past 2 years, it still has its limitations due to the relatively lower editing efficiency and also with limited vendor of GMP production. But now it's become more promising and more popular in the field. To do the replace the endogenous TCR with antigen-specific TCR and CAR, first we need to design a special guide RNA to create double-strand DNA break at the first exon of TCR alpha constant, and then integrate the design constant at the double-strand break via homology directed repairing. In this way, TCR or CAR expression cassette can be placed under the control of endogenous transcription regulation, leading to a sustainable T cell function and delayed cell exhaustion. On the left side of this slide is a detailed design of how to insert a NY-ESO TCR to the endogenous TCR to generate TCR-T. While on the right side of the slide is a design how to replace endogenous TCR by a well-designed anti-CD19 CAR. To do the T-cell engineering via non-viral protocol, we need a few key components. First of all is the guide RNA, and it needs to be mixed together with Cas9 protein to form the active form of RNP. And we also need the HDR template to introduce the CAR or TCR constructs. Also, we need the isolated and activated T cell. When all the materials are ready, we mix them together and put on the electroporation platform, and then do the cell expansion and recovery, and to screening the positive population. To achieve high editing efficiency, the choice of RNP materials is very important. Let's go back to the case of PD-1 knockout. Here are 3 different published data. On the first one was a clinical trial done a few years ago by a Chinese group. They delivered Cas9 guide RNA via plasmid templates. The expression of Cas9 protein and guide RNA were all done by in vivo transcription, and the average knockout of PD-1 is only about 10%. And back to the case that we mentioned before, that report in 2020 for the knockout of PD-1 here, the guide RNA is actually produced by in vitro transcription, and the knockout efficiency of PD1 is averagely in 20%. On the right side is the recent case published in the end of last year. It's actually also a Chinese therapeutic company. They developed a new cell therapy by deactivating the endogenous PD1, meanwhile knocking a anti-CD19 CAR at the PD1 locus. In that study, the researchers used a highly purified single-guide RNA, which is chemically synthesized, and it's actually purchased from GenScript. The knockout efficiency of PD1 is about 90%. The other important thing is to ensure the material for knock-in templates. We did some in-house tests to verify which is the best form for knock-in templates done on hectonecary T cell line. We choose a few different types of payloads as represents like plasmid DNA and mini circle to represent circular DNA payloads, and the purified PCR amplicon to represent the blunt and linear double-strand DNA, and also a single-strand DNA. 2 days after electroporation, we found both plasmid DNA and the PCR amplicon has quite high GFP positive rates, but we also found very high background with a non-RMP control group, which means this is actually transient expression of the constructs. When we look at the ssDNA group, the GFP positive rate is pretty low, but there's also barely any background in the non-RMP control group. When we wait till 7 days after electroporation, we found out at the circular DNA payloads group, the RMP positive samples has much dramatically decreased GFP positive and almost equal to a non-RMP control group, which means there's no knock-in events happened. When we look at the PCR dsDNA group, the GFP positive rate maintains at about 20% and is much higher to the control group, which indicates there's actually integration happened. For single-strand DNA, the positive population doesn't change, and also in the non-RMP control group is still with very low background. So the conclusion here is linear DNA payload templates have better performance than circular DNA templates for HDR. And at the same dosage, like 2 microgram, single-strand DNA has lower knock-in efficiency, but also lower false positive signals. In the end of 2019, there's also a research group published work by using long-read sequencing to evaluate different types of payloads like what we did. There are PCR donors, which is linear, blunt, and double-strand DNA. Also single-strand DNA donors and plasmid donors. By long-read sequencing, they found that, by FACT evaluation, they found that, like what we just reported before, that PCR donor gives the highest GFP positive rate with like about 50%, while single-strand DNA donor gives 20% GFP positive rate, and plasmid DNA gives the lowest GFP positive rate. When looking inside the constructs by long-read sequencing, they found out besides the perfect HDR insertion, there are also a high ratio of duplicate NHEJ ligation happened when using blunt and PCR products. And also integration of concatenator donors happens when using PCR amplicon as HDR repair templates. Well, for single-strand DNA donor, there's much higher ratio of perfect HDR, and there's barely any other type of random integrations. Well, however, there can be or might be low ratio of truncated integration donor due to maybe the single-strand DNA is not stable within the cell. And the case for plasmid DNA donor is the worst. It has much lower ratio of perfect HDR, but also have other risks of with the integration of extended plasmid backbone. And here we also have some in-house testing on dsDNA we purified from a PCR amplicon compared to single-strand DNA donor on a primary T cell from acute ALL patients. On this data, we found that double-strand DNA will cause severe cell death due to toxicity via a dose increased manner. Well, for single-strand DNA, it can, with the increase of the dosage, the survivability maintained at a all very high level. And most importantly, when we look at the effects data, and the 4 microgram dosage, there is no positive population anymore because most of the cell died due to toxicity of double-strand DNA. However, for single-strand DNA, the knock-in efficiency increased linearly with the dosage of siDNA input. Also, by the very beginning of 2021, there's also another German research group reported the toxicity of double-strand DNA is actually dependent on the dosage of transfected double-strand DNA. It can trigger multiple autoimmune response, which may lead to cell death or apoptosis. In this research, the authors also combined the DNA sensor inhibitor and also with HDI enhancer, which can give a very nice protocol for the cell preparation and cell treatment electroporation program. In the end, by combining DNA sensor inhibitor and HDI enhancer, the knock-in efficiency of anti-CD19 CAR and the BCMA CAR can both increase from 20% to about 50%. The take-home message is that T cell can be edited efficiently by non-viral methods. High-quality chemical synthesis single-guide RNA can provide high coding efficiency and low off-target rates. Better knock-in efficiency, cell viability, and integrity can be achieved by using single-strand DNA payloads as repair templates and optimized electroporation protocols. And in the end, I would like to introduce GenScript's CRISPR service supporting gene knock-in, including the CRISPR sgRNA service and the single-strand DNA service. For single-guide RNA at GenScript, we offer highly purified, safe-edit sgRNA. It has minimal impact on cell viability and minimal off-targets from truncated oligos, and it's one-stop shop from CRISPR machinery to knock-in templates. Here's some data to show the high purity of our highly purified single-guide RNA. It has a purity more than 99%, and there's no other truncation fragments in the final product. Also, it has lower toxicity compared to IVT prepared single-guide RNA due to the lack of triphosphate at the 5' end. And also because it higher stability due to the modification at both 5' end and 3' ends. And also it has very high editing efficiency and safety on primary T cell. As you can see here, with both Cas9 and Cas9 fused with EGFP protein at different dosage from 7.5 picomole to 20 picomole, we can all reach better than 95% of the knockout efficiency and high cell viability around 80%. Also, we also provide single-strand DNA service for the HDR repairing templates. The single-strand DNA we produce is done via our proprietary isothermal enzymatic production process, and also is produced by our in-house prepared high-quality controlled starting materials. The length is available from 150 to 500 nucleotide long. And now we are glad to announce that it's up to milligram level delivery quantity, allowing for flexible study design and even to support preclinical studies. The dsDNA final product, single-strand DNA final product is sequence verified, both of the DNA producing template, also the final ssDNA product. And we also offer ultra pure grade for large scale single strand DNA, and it's animal free and endotoxin free. And we also offer free lifetime gene template storage supporting faster and more cost-effective reorders. And in the end, I would like to say that we are the expert in gene synthesis with 18 years experience, and we can synthesize more difficult genes as also more difficult single-strand DNA designs. Also, for longer designs and DNA payloads, we also offer milligram level linear stable dsDNA as knock-in templates. In the end, I would like to say, let's work together to improve the efficiency and the safety together. And thank you so much for listening. If you have any questions, I would like to, I'm happy to answer. If you have any further questions, please visit our website. Thank you very much.
Thank you, Lumo, for the great talk. Please enter your questions in the chat box and we'll get to it via chat or at the end of the session. And now let's move on to our next talk.
Hello, everyone, and thank you all for attending today's webinar. My name is Koming, Global Product Manager at GenScript, and I have the pleasure of introducing our speaker today, Dr. Joshua Burgess. Dr. Burgess graduated with his Ph.D. in 2016 from the Queensland University of Technology. He is currently an Advanced Queensland Early Career Research Fellow working within the Cancer and Aging Research Program at the Queensland University of Technology. Dr. Burgess's work seeks to determine how genome instability contributes towards the development of cancer. Specifically, he is studying the function of key DNA repair proteins in order to aid the development of new drugs and biomarkers for cancer treatment. Today, Dr. Burgess will share with us his efforts in studying a new molecule that plays a role in DNA damage repair. He will also describe the potential in using this molecule as a companion diagnostic to help identify cancer patients most likely to benefit from PARP inhibitors. With that, I would now like to pass the floor to Dr. Burgess.
Thank you for the introduction. As mentioned, I'm a member of the Cancer and Aging Research Program here at the Queensland University of Technology. Today's talk is titled Characterization of PARP inhibitor biomarkers for cancer therapy. So our lab has a core focus on DNA damage repair, and this really provides a foundation for all our research projects. This feeds into our cancer-focused work, where our lab is focusing on currently a phase 1 clinical trial for a new cancer therapy. As well as this, we're developing a new biomarkers, which is the focus of my study, and this is really to have the driven goal to improve patient survival. As well as this, the DNA repair studies has also taken us to look into aging, with really a goal of increasing the healthy lifespan by reducing aging-related diseases. So this is things such as Alzheimer's. We do this through the development of new novel compounds. As I mentioned, the core of our lab is DNA damage repair, and there's a multitude of types of DNA damaging events. And this then requires different DNA repair pathways to repair this damage. So for example, here you can see bulky adducts, which are repaired by the nucleotide excision repair pathway, whilst you have base mismatches, which are repaired by the mismatch repair pathway. We have double-strand breaks, which are considered the most deleterious type of DNA damage and can be repaired by either non-homologous end joining or homologous recombination. And finally, single-strand breaks, which are repaired by base excision repair. Our lab actually has a primary focus on double-strand break repair, and here you can see when the DNA is damaged, it can be repaired through homologous recombination. So this is where the DNA is first initially recognized by the mRNA complex. This then allows the resection of the DNA, resulting in single-stranded DNA. Then RAD51, this important DNA repair protein, then can bind this single-strand DNA, where it is then able to facilitate strand invasion. So it uses a homologous DNA sequence in the genome as a template for the repair. But this then means the requirement of a homologous template that these can only function with the G2 and M phase of the cell cycle. The alternative pathway, non-homologous end joining, is somewhat more simplistic, which is simply the recruitment of DNA repair proteins, then religation of the broken ends. This, however, has a higher risk of loss of genetic information, as no template is used for the repair. A similar process can be seen within the other repair pathways, with basic steps of DNA damage recognition, followed by the recruitment of additional DNA repair proteins, then the excision of any sort of DNA base or nucleotide, and finally the synthesis of new DNA and the religation of the damaged DNA back together. So here I'm really just indicating how the DNA is actually targeted in a number of cancer therapies. So for example, platinum-based chemotherapy, which results in DNA crosslinks, whilst topoisomerase inhibitors cause both DNA adducts as well as DNA double-strand breaks, and radiation causing direct DNA double-strand breaks. So these compounds actually really exploit the cancer cell's vulnerability to genomic instability and their really high replication rates. So this is really designed to specifically target these cancer cells. Chemotherapy patients, however, actually often experience a lot of toxicity and detrimental side effects due to the impact of these chemotherapies on their healthy tissues. So really the focus of my study is on a protein called SASH1, or salmon SH3 domain containing protein 1. So SASH1 has previously actually been described as a tumor suppressor. So this means it really has reduced expression in cancer. This has been shown within breast, lung, thyroid, ovarian cancer, as well as colorectal cancer. The reason we began working on SASH1 is after some actual bioinformatic correlations, where it was identified to be inversely regulated with some key DNA repair protein genes, which we had a particular interest in our lab. So previous work on SASH1 has identified it to have roles within apoptosis, where it's actually cleaved by caspase-3, and this has been linked to its role in NF-kappa B signaling pathways. Additionally, it's been shown to affect the PI3 kinase pathways, as well as the actin-E cadherin structures. The schematic here that I'm representing is of SASH1 protein, and as the name suggests, you can see it has a number of SAM SH3 domains, and these have previously been shown to be involved in protein-protein interactions. In addition to this, it has nucleolocalization signals, which would be expected of proteins that require directly in DNA repair to be localized into the nucleus. As we worked on the fundamentals of DNA repair, and we initially identified SASH1 to be correlated with known DNA repair genes, we first wanted to look to see, does SASH1 actually have a role in DNA damage repair? So to assess this, the first result I show here is the neutral comet assay, which directly assesses the amount of DNA damage in a cell. So here you can see the longer the comet actually indicates more DNA fragmentation or more genomic instability. This is because an electrical current is placed across the cell, and this is whilst they're embedded in agrose, allowing the DNA to separate based on size. So the more fragmented, the longer the DNA. And in this case, actually depleted SASH1 from cancer cell lines using sRNA. And we can see that this actually presented a severe increase in DNA damage events, as you can see by the fragmented DNA longer comet tails. This was really the first evidence we had to show that SASH1 may actually play a role in DNA damage repair, as well as genomic stability. Subsequent to this, I looked to see if the protein was required for the repair of DNA damage. So to look at this, I looked at the repair of DNA double-strand breaks through a DNA damage marker gamma H2X, which is a modification of a histone at DNA damage sites. And as you can see, this can be done through immunofluorescence, where each DNA double-strand break is marked with a gamma H2X signal. So following the induction of a DNA damage using ionism radiation, once again in control in SASH1 depleted cells, we can immediately see a marked increase in foci count. Then over time, we can see this decrease, indicating functional repair pathways. However, when we depleted SASH1, we can actually see that this repair process is delayed. So this indicates that SASH1 actually may be required for the direct repair of the DNA. The next experiment we undertook was an experiment called a homologous recombination assay. So in this assay, we looked directly at homologous recombination by inducing a specific ISC1 double-strand DNA break. So in this case, after the DNA double-strand break, if it's repaired by homologous recombination, it actually results in a functional GFP protein in a green cell. And using this, we can measure it by flow cytometry. Once again, when I simply deplete cells of SASH1 using sRNA, we can see an impaired homologous recombination efficiency. So really, based off these results and a few others that I haven't gone through, we hypothesized that cells lacking SASH1 may be unable to repair cells of DNA damage events, and actually might be sensitive to some current chemotherapy aids. But in particular, we suspected that SASH1 may be sensitive to PARP inhibitors due to synthetic lethality. So what is PARP inhibitors and what is the synthetic lethality? But really, targeted therapies are often associated with a more favorable patient outcome, given there's significantly less of off-target side effects. So a more directed treatment approach has been coined as synthetic lethality, and the example here is PARP inhibitors. So normal cells following DNA damage events, they can be repaired, in this case by the well-known breast cancer-associated genes which are involved in multiple recombination, being BRCA1 and BRCA2. But in cancers, often the BRCA genes are mutated, resulting in reduced DNA repair capacity. And this then makes the cells reliant on alternative DNA repair machinery. In this case, you can see this is through PARP. So when BRCA1 is lost, the cancer cells are then necessary to use the PARP machinery to repair these DNA damage events. But we can actually exploit this vulnerability by using PARP inhibitors. So you can see here, we use a PARP inhibitor to inhibit PARP, it actually results in targeted cell death. So normal cells, when you inhibit PARP, if they have BRCA, they'll able to repair this DNA damage. But now the cancer cells that don't have BRCA will actually be sensitive to this DNA damaging agent, resulting in more selective tumor targeting treatment. So in our case, we were proposing potentially SASH1 levels might function similar to BRCA and predict which tumors might be responsive to PARP inhibitors. So looking what is currently out in the market now for PARP inhibitors, they use BRCAness, so the mutation status as well as the functionality of BRCA genes, as well as the HDR status as their biomarkers. So the HDR status can be measured through a few different means. So this is such things as mutations within additional homologous recombination genes, or single nucleotide polymorphisms, and then as well as the phenotypic or functional assays of the genes. So outlined here, these tests can be from a number of sources, both being germline and tumor testing. But with the germline BRCA mutations, this actually, when the mutation is found, actually significantly increases the risk of developing breast and ovarian cancers. So you can see here, the risk of breast cancer drastically increases from up to 60% in women who inherit these BRCA mutations. So I just wanted to highlight some of the mechanisms behind PARP inhibitors so you can appreciate what their functionality is. So there's actually been a few proposed mechanisms, and I'll briefly touch on them. So the inhibition of PARP has been proposed to inhibit PARP's function in the repair of single-strand DNA damage. So PARP is actually required for the pyrrolation of proteins for the successful repair of the DNA lesion. However, some studies have also demonstrated that there isn't a massive increase in single-strand DNA breaks following PARP inhibition, as well as through the depletion of PARP1, once again using siRNA. So this doesn't have the same toxicity as the inhibitors, so there's some question there might be additional mechanisms involved. So an alternative explanation has been stalling replication forks. So the PARP1 is involved in the required for the restart of replication forks. So if we inhibit or deplete PARP, the replication forks end up collapsing, which then requires homologous recombination to repair this now forming double-strand break. So therefore, in the BRCA-deficient tumors, these would be unable to repair this DNA damage and can result in the targeted cell death. PARP trapping has also been a focus of a number of PARP inhibitors. So this is where PARP has been stuck on the DNA from the inhibitor binding. This then creates an obstacle for the replication, resulting in the replication fork hitting this bound PARP and causing a DNA double-strand break, once again requiring homologous recombination to repair this lesion. There are some data to support this theory, however, unfortunately, it hasn't quite translated into clinical findings, with the best PARP trapping inhibitor not always having the best clinical benefit. As well as this, due to PARP's role in replication through its pyrrolation of proteins, this is for both the activation and deactivation, it has been proposed without this mechanism, the replication isn't controlled correctly, so this actually results in faster replication speeds, therefore generating more single-strand breaks in the genome. And this accumulation of single-strand breaks can actually lead to cell toxicity. Another mechanism proposed where the PARP inhibitors actually alter the expression of transcription of proteins. So in particular, PARP1 has been associated with the activation of E2F1, where it's a transcription factor for a number of DNA repair genes, as well as a number of screening studies have identified factors such as P53 and FKB being altered following the inhibition of PARP. So this would result in further defects in the DNA repair machinery and more toxicity for the cells. The final proposed mechanism is actually an increase in non-homologous end joining promotion, so the alternative DNA double-strand break repair pathway. The PARP inhibitor treatment has been shown to increase the phosphorylation of DNA PK substrate, the main protein involved in non-homologous end joining, and this consequently promotes non-homologous end joining. So in combination with the loss of recombination in the BRCA mutant cells, this would force cells to use the more error-prone mechanism of non-homologous end joining, which once again accumulates the DNA damage in these cancer cells, resulting in a cell death. However, from all these proposed mechanisms, you should be able to appreciate that there is no clear function, and it's more than likely that multiple of these mechanisms are actually contributing to the toxicity seen within these cancer cells. If we look at the current status of PARP inhibitors, you can see that 3 have been approved for the treatment of ovarian cancer patients with BRCA mutations, being olaparib, ruxolitinib, and talazoparib, in addition to niraparib being approved for the treatment of homologous recombination deficient ovarian cancer. One major caveat of this treatment option is actually they require an active biomarker for their treatment, with I mentioned before being the BRCA and HDR status, but this is actually only selecting about 30% response rate. So there's a large proportion of patients which are receiving these treatments who aren't actually responding. And with really a continual growth in the use of PARP inhibitors, it makes sense to identify better biomarkers to have better patient outcomes. So for my project, this is sort of the basic outline we were looking at to develop a biomarker. This can be quite different for different projects. But the basic steps we have been doing is initially discovery phase. So this is the basic research. So basically screening to identify a potential biomarker. And this is how we identified SASH1. And through this phase, we also wanted to get a good detection system. So this is where we optimized to show that it was actually the protein of SASH1 we had to look at, not the mRNA. Next, further validation would be required. So depending on the biomarker, we're actually looking at cell lines as well as PDX samples initially. And then we would really want to provide a foundation to support the efficacy of undertaking clinical trials. And for the clinical trials, initially we're trying to get retrospective studies. So this would really be great to utilize if possible, so to save us having to go through the expensive process of undertaking our own clinical trial. And really with the main goal of validating the use of the biomarker and showing it's better than the current clinical practice, being the BRCA or HDR status. And if all was successful, then we would move into the approval phases. So the benefits of PARP inhibitors has actually been quite promising so far, but as I mentioned, only 30% of BRCA mutant tumors are actually responding. But this statistic's actually been quite variable, with up to 50% in some studies and as low as 20% in other studies. This has been primarily based off the difference in criteria of what they classify as being responsive or not responsive. But looking at the benefit in survival, we can see that one breast cancer study showed an improvement of survival from 4.3 to 11.2 months. This was in the BRCA mutant cohort, whilst the BRCA wild type cohort only showed an increase in 2 months survival. Similar ovarian cancer studies have also shown a 13.9 to 49.9 survival with the BRCA mutant, so quite a large survival benefit with the treatment of PARP inhibitors, whilst still quite a decent increase in the BRCA wild type with a 5.5 to a 19.1 month improvement. And it can be seen that there's actually a good proportion of patients who are BRCA wild type that are responding to this treatment. In particular, studies with additional PARP inhibitors and niraparib and ruxolitinib have shown this. So really, we would want to try and identify these patients with other biomarkers. And here, I'm just showing a couple of Maya plot indicating how good some of these PARP inhibitors have been. So this was an olaparib and placebo-treated cohort with BRCA mutant patients only. And you can see a massive increase in the survival benefit of these patients. So my research is leading to the development of a new PARP inhibitor biomarker, which we're proposing to be SASH1. So I'm going to show you some of the data I've generated for this part of the project. Really, the real reason we need to do this is because currently only 2% of breast cancer and 10 to 15% of ovarian tumors present with this BRCA status. And with only a 30% response rate in this cohort, there's really only a small proportion of patients who are currently being able to benefit from this treatment. So it's clear from a number of studies that agents can actually be beneficial beyond this small niche of patients, and to make it become more widely accessible to other cohorts, a new biomarker would need to become available. So the product we're sort of developing is a new monoclonal antibody against SASH1. So So to secure this, we've actually got a patent on our target and our approach. And to date, we've tested our biomarker within cell line models and PDX models. And we're currently working on getting retrospective clinical trials. It is also quite important that we actually have the resources through our lab as well as collaborators to continually push this project further forward. It's something we really consider when undertaking projects like this. So to initially investigate this, I utilized panels of cancer cell lines. So here I have breast, lung, and ovarian cancer cell lines. And each dot on these graphs represent one cell line, where I've assessed both the expression of SASH1 as well as sensitivity to the various PARP inhibitors. And it can actually be seen in all cases, there's a strong correlation with low expressing resulting in low IC50, therefore more sensitive to the PARP inhibitor. This is really what we've predicted, is when SASH1 is low, there's a defect in DNA repair, and we would predict that they are sensitive to the PARP inhibitors. This was seen with the multiple number of PARP inhibitors, so I've used olaparib, talazoparib, and olaparib in the breast cancer panels, as well as we've done multiple cancers, so we have the breast, lung, and ovarian cancer. So potentially, SASH1 could be quite a broad applicable biomarker. So to do some validation of these findings, actually depleted the protein again from the cells. This was once again using sRNA, and then calculated the IC50 to olaparib. And it would be hypothesized that when you deplete the protein, you would actually increase the sensitivity to PARP inhibitors. And this was seen once again in both the lung and breast cancer cell line panels. In addition to this, I wanted to stably overexpress the protein to see if I could make the cells more resistant to PARP inhibitors. And indeed, this was the case. As you can see in the third graph, the increasing SASH1 expression actually made the cells more resistant to PARP inhibitors. So far, this data is actually lining up quite nicely. The SASH1 may be a potential biomarker for PARP inhibitors, but this has only been undertaken within cell line models. So we really need to progress beyond cell lines. So initially, we looked at doing some in-house PDX investigations. So here we can see a panel of PDX samples, which I've checked by Western blot, the SASH1 expression. And from this, we were able to select one low and one high PDX. And importantly here, you can see on the right the table, we also actually scored this by immunohistochemistry staining. So we wanted to make sure that this antibody and this biomarker was applicable within more of a clinical setting, because they're not going to do the Western blots within the clinical practice, but IHC is a standard protocol. So from this, we selected the LP28 to be a low expressing SASH1 PDX and the G0183. And then we treated them with the PARP inhibitor olaparib. And when we measure the tumor rate following treatment, we could see the low expressing SASH1 LP28 actually had a significant response to PARP inhibitor treatment at both the low and high doses of olaparib. Alternatively, the high expressing SASH1 PDX, G0183, had quite a poor response, only just reaching statistical significance at the highest dose. So this is really what we were expecting. But we needed more data than 2 samples. So we got additional retrospective PDX panel, which had known PARP inhibitor sensitive data from a collaborator at WeHi, Claire Scott. And as you can see here, there was a number of PDX samples, so 24 samples we've received so far. And when we were actually blinded to this data, which is quite important, we weren't aware of the sensitivity prior to testing the expression of SASH1. But when this was revealed, we could see that the sensitive cohort actually had significantly less SASH1 expression than the refractory cohort. So once again, supporting that low SASH1 would predict the sensitivity to PARP inhibitors. Additionally, it should be noted, this was regardless of BRCA status, with a number of BRCA mutant samples actually having high SASH1 expression and not being responsive to the olaparib treatment or reciprocal treatment. So then we wanted to move back into our cell line assays. So once again, I utilized the common assay to look to see what's going on following a PARP inhibitor treatment, in this case olaparib. And once again, we can see when we depleted cells of SASH1, we get an immediate increase in comet tail length. But following the PARP inhibitor treatment, the control cells also have a marked increase in DNA damage, marked by the longer comet tail. Interestingly though, the overexpression of SASH1 actually resulted in a reduction in DNA damage from the PARP inhibitor. So this could suggest that the protein is actually helping to either repair the DNA damage or it could be preventing the DNA damage in the first place. To look at this in another way, I actually looked at a panel of ovarian cancer cell lines endogenously. So this is without altering the expression. And I separated the cells both based off low and high expression of SASH1. Then looked at the DNA damage marker RAD51. So this is similar to the gamma H2X foci, which I showed earlier in the talk. And we can see that I treated cells with olaparib and then allowed them to recover for 24 hours after treatment. But you can see the difference between the low and high expressing cells, where the low cells were unable to return to the control levels of RAD51 after the recovery, whilst the high expressing cells actually able to have a marked decrease, getting closer to that control RAD51 expression, indicating they were able to repair the damage. So this really provides some further evidence that SASH1 is required for the repair of DNA damage, in particular in PARP inhibitor-induced DNA damage. It was clear at this point in the project that we needed some clinical applicable data to progress further. We actually found it quite difficult to find any available TMAs with PARP inhibitor response data as well, as the treatments of PARP inhibitors are still relatively recent and a lot of the studies are still based within clinical trials. So initially we just simply looked at the expression profile of SASH1 within cancers. So this was done to identify different staining patterns, as well as the prognostic implication and what proportion of patients may actually be treatable with our PARP inhibitors based off the SASH1 expression. So here I'm showing our ovarian cancer TMA we undertook. This was composed of 96 patient samples, and the staining was seen to be both nuclear and cytoplasmic, but the nuclear intensity actually correlated patient prognosis. But interestingly, this was different to the previously published data where SASH1 has been described as a tumor suppressor, as the positive tumors actually had a poorer prognosis than the negative cases. We believe this is primarily based off the previous studies being from mRNA, and we have shown in our lab that the mRNA and protein don't have strong correlation. Interestingly though, from this study, we were also able to determine that 42 of the 96 patient samples had no detectable SASH1. So we would propose that these tumors would actually be the best cases to treat with the PARP inhibitors. So we undertook a few TMAs, and this is the summary of the findings. So important findings were that there was decrease in expression in the majority of cases compared to the adjacent normal tissue. A subset of tumors had no detectable marker. So this was 25% in breast cancer, 10% in lung, 40% ovarian cancer. And this is markedly above that of the BRCA. So BRCA status is only seen in 10 to 15% of ovarian cancer and 5% in breast. And interestingly, this may also increase the ability of introducing PARP inhibitors into a larger proportion of lung cancer treated patients. We did, however, identify a number of issues while undertaking this part of the project. So we were utilizing commercial antibody for the project up to this point, and we showed it was quite inconsistent between lots within immunohistochemistry staining. And we had the TMAs done at multiple institutes, and then it required a lot of optimization between each set of samples. So to progress this project further, we really couldn't be reliant on this sort of system. So we needed to make our own antibody in case this commercial antibody ends up becoming not produced, and then it would actually make our approach unviable. So to do this work, we actually began a project with GenScript to develop our own antibody. So this is the basic outline of the process we undertook. So we provided 7 milligrams of protein to GenScript to start the project. We actually decided to use full-length SASH1 for this. It gives the best chance of getting strong antibodies. In our case, we already had the protein being produced, but GenScript is also able to produce the antibody for you if you didn't have this system. Following this, GenScript immunized 4 rabbits with our protein to develop antigens against the target. Following these immunizations, ELISA tests were performed at multiple stages by GenScript, and all this data was provided to us. In addition, the bleeds were sent to us following all the immunization steps, and we were able to screen these in-house to identify which rabbit was best to proceed with to develop new antibodies. And from this, we decided which rabbit to get monoclonal hybridization and screening done. And then we were given 10 top candidates, which were then sequenced and small batch purification was performed. And these antibodies are shipped to us for validation and selection. In addition, GenScript did additional ELISA tests to identify which of the antibodies had the best reactivity. And from this data, we were able to select one or even multiple antibodies for which we wish to have large-scale purifications. But in addition to this, we got the antibody sequence report and we got plasmids produced. This allows us to then produce the antibody in-house. And really the reason we went down this path was to make sure we didn't have to redo any of our clinical data with the new antibody in the future. So I just have some very summary data showing that we've actually received our 10 clones. And from this, 4 worked very well in Western. So the first blot here, I'm showing simply SASH1 expression or SASH1 depleted cells with the commercial or clone 10 in this case, showing we can detect the antibody, detect the protein well in the non-depleted sample, whilst the depleted sample we see loss of SASH1. Additionally, we produce cell pellets to confirm by immunohistochemistry. In this case, had control or SASH1 overexpressing cells. And once again, we can see that the clones, as well as the commercial antibody, detected SASH1 expression. And additionally, we had the immunohistochemistry performed on small TMAs. So this was really done to make sure that the localization and the staining pattern of both the commercial and clone antibodies were similar to get the best clone for our applications. And what we could see that there was clear comparison between the staining with predominantly nuclear staining of SASH1, as we've seen previously, as well as the high expressing PDX, the TMA samples having similar high in both commercial and clones, whilst the low expression having low both in the commercial and the clones. In addition to this, we were actually given the plasmids to produce our own antibody. So I wanted to make sure that we could do this in-house as needed. So to do this, I actually overexpressed the plasmids in HEK cells when then the antibody is then exported into the media. I then ran this media over a protein G column. So you can see here, this is a readout of protein going over the protein G column. And initially you can see the unbound protein is coming through the column, indicated by the large first peak. And then we washed the column and then eluted the antibody using glycine and a low pH, which is the second and smaller peak. And we can confirm we got the antibody by running chromatic stain, where we can see both the heavy and light chain as expected from an antibody. As well, you can see the concentration of the antibody. You can even detect it within the input sample. But the main point is, does this antibody we produced ourselves actually work? So once again, I just did a simple Western blot. In this case, I had control or SASH1 overexpressing cell pellets, and we can see the antibody easily picking up the overexpressed SASH1. So a summary figure of what we've been proposing. So in tumours that are deficient of the biomarker, they would have impaired DNA repair, and this would then allow them to respond to PARP inhibitors, whilst the positive SASH1 tumours would have a poor response, and really alternative treatments would be preferable for these patients. So what are we up to in the near future for this project? Well, firstly, we're actually trying to develop a training set of samples to maintain continual quality control over our antibody. So when we move into a clinic, they'll have samples that run alongside the patient samples, have positive and negative staining to ensure that the staining is consistent and the outcome is well controlled. In addition to this, we're accessing both retrospective and new patient samples, so moving beyond just PDX samples, which have been treated with PARP inhibitors. This is really needing to expand our data to progress it into more of a clinical trial application. In addition to this, we want to move beyond ovarian cancer samples and start to collect samples from other cancer types. This is really to assess is SASH1 a more robust biomarker and can be used for a bigger cancer inhibitor therapies. But this just finally leads me to acknowledge the Cancer and Aging Research program, as well as the funding, in particular from my Advanced Queensland Fellowship, as well as the National Breast Cancer Foundation, which have both directly supported this project.
Thank you, Dr. Burgess, for the wonderful talk. Before we end, I would like to take this time to share a bit about GenScript's reagent antibody services. To date, we have successfully completed more than 15,000 antibody projects of various complexities. Recently, we have upgraded our rabbit monoclonal antibody generation workflow with the introduction of B-cell cloning, bringing you higher clone numbers and diversity. Moreover, we are now having a 20% promotion on all monoclonal antibody projects when you start one with us before June 30th. For more information, please visit our website or email us at antibody@genscript.com. With that, I'd like to thank you for your attention. Please feel free to enter your questions in the chat box and we'll get back to you if we do not have the time to address them immediately.